Professor Grant's, Dr. Grant's got a PhD from the University of McGill in pharmacology and therapeutics. He did a postdoc at the Geisinger Medical Center for Research in Pennsylvania, where he studied extracellular calcium sensing receptors. He did a postdoc at the LDI, where he investigated the role in calcium sensors in musculoskeletal disorders. He's currently a research associate in the laboratory of Doctors Moali and Antonio. In addition to calcium receptors, he investigates the effect of heavy metal exposure on the development of osteoarthritis and chronic back pain, and the peptide link N. He's developed a media system which you can buy from WeSend Bioproducts. He's involved in chitosans for the treatment of chronic back pain. He owns a startup company called Intervertech and consults for a couple of companies in the biomechanics space. So the title of his talk today is Calcium in the Extracellular Calcium Sensing Receptor in Musculoskeletal Disorders. Welcome, Michael.
Thank you. Thank you for the introduction. So if we just go to the next slide. Yeah, sure. So calcium homeostasis, very important part of regular physiology. It is highly and tightly regulated. There are several organs involved. We have the bone that's involved in calcium resorption and deposition. You have the gut that's involved in also uptake, and the kidneys are involved in resorption and excretion. The hormone, the central hormone, well, there are two, but the principal hormone is the parathyroid hormone, and that hormone acts on those various organs, and these organs would be considered the calcitropic effects on calcium homeostasis. The other hormone that balances the calcium, serum calcium levels is calcitonin. So parathyroid hormone increases the serum calcium levels, calcitonin reduces them. Next slide. Yeah, so the principal receptor that's involved in regulating parathyroid hormone secretion from the parathyroid glands is the calcium sensing receptor. It's a G protein coupled receptor. And like many of the G protein coupled receptors, it's been shown to function as a homodimer. And what's unique about this particular receptor, it's quite large, and the large part of it is the extracellular domain. And this extracellular domain operates almost like a venous flytrap. It's been shown by crystallography. And calcium essentially binds in the lobes of these of this venous flytrap, and then upon calcium binding, the lobes close. This induces a structural change to the receptor, which leads to signal transduction. Next slide, please. So this is it. So there are over 1,000 residues that make up the calcium sensing receptor. And what's actually compared to other receptors, and this makes it again a little bit unique, is that there have been over 300 variants reported. And many of these variants have been shown to have a function, a disease aspect to them. The majority cause this loss of function, called loss of function mutations, and that's where you have reduced or no activity from the calcium sensing receptor. This leads to a hypercalcemia. The other mutation, less common but also prevalent within these variants, is the gain of function mutations, and in that case, increased activity of the receptor, you have decreased parathyroid levels since it regulates parathyroid secretion from the parathyroid glands, and that leads to a hypocalcemia effect. Next slide. So the regulation of parathyroid and calcium or calcemia in in our blood is considered the calcitropic actions of the calcium-sensing receptor. And like I mentioned, so these activities occur on the central organs, the kidney. It has an effect on bone, gut for absorption, and the mammary glands. So particularly during lactation, calcium levels are sensed, decreased calcium levels are sensed within the mammary glands, and this induces resorption from the bone and increased calcemia, which replenishes the calcium stores in the lactating mammary. In the kidney, it could go typically depending on what part of the kidney, because it is expressed in different regions of the kidney. It could act as a increased resorption or decreased resorption. Next slide. So the calcium sensing, I'm not going to go into detail on all the signaling pathways that are elicited by the calcium sensing receptor. There are several. One thing about it, like I mentioned, it's a G protein coupled receptor, so it does couple to G proteins. And the other aspect is that it falls under the category as a promiscuous receptor, in the sense that it can couple to G alpha I, which inhibits cyclic P activation, and G alpha Q, which activates calcium stores and increases calcium levels in the cell. Those are the things that were known. What was recently discovered, and that's something that I worked on in a postdoc, was that we discovered that in addition to these aspects, the receptor itself also is regulated differently compared to other GPCRs or receptors in general, in the sense that agonist doesn't induce a decrease or desensitization or downregulation of the receptor, as often is the case for other cell surface proteins, but in this particular case, it induces an agonist-driven insertional signaling. So if you go to the next slide. So what we found was that using an engineered receptor with a GFP attached to the extracellular domain, and this GFP, the one that we use, is called fluorine. The reason why it's called fluorine is a pH regulator. So when this receptor is expressed intracellularly, which is typical intracellular expression, the environment is often acidic. The fluorescence of this receptor is markedly reduced. So the fluorescence of the GFP tag that's on the extracellular domain is markedly reduced. But upon membrane insertion, plasma membrane insertion, and exposure to the extracellular milieu, where pH is often regulated at 7.4, the fluorescence is reinitiated, and you could do your fluorescent microscopy with that. In addition, we also inserted at the N-terminus, just before this fluorine GFP, was called a bungarotoxin sequence. And what was unique was that the bungarotoxin is a very small peptide, and what you can do is you could purchase them with different fluorophores attached to them. So now the reason for this was we were able to label the receptor in two colors. For the bungarotoxin, which I'll show later, we label it with red, and the GFP, or the fluorine conjugate, was obviously green. So if you see here on the top panel, what we've noticed when using a total internal reflection microscopy, so this is TIRF-M microscopy. Again, a unique property of TIRF is that you're literally just seeing what's present on the cell surface. You could adjust the angle of the laser, and you're capturing light, emission light that's coming directly from the cell surface. And what we found was that in low calcium concentration, this is 0.5 millimolar, you saw a receptor expression, but when we increased the calcium levels in the bath, we found a marked increase in membrane insertion or expression, and this was rapidly decreased when you reduce the calcium levels back to normal, or 0.5. And present on the bottom left hand, you see that with increasing, these curves in blue represent increasing calcium concentration. So the higher the calcium concentration, more insertional or more membrane insertion of the calcium sensor was observed. And we also did some studies to show that the membrane insertion was directly associated with signaling. So one of the, like I mentioned before, that the calcium receptor promiscuous, it could also activate intracellular calcium stores through G-alpha-Q coupling, and using the fluoro-red dyes, we could see that this signal, becomes saturated as you increase the calcium in the media. And that was directly correlated in the top graph, which is in black, in the bottom right hand. So the top bar, you see as you increase calcium, you also increase the membrane insertion of the receptor. The next slide. And finally, we demonstrated that this insertion of the membrane in the membrane was not due to a recycling event. It was mostly due to actual recruitment of newly synthesized receptors that were kept in a storage in the ER. And I don't know, Misha, if you could actually activate. I'm not sure if that's
no.
Probably not.
Oh, sorry.
No, it's fine. So what happens was that as the red, when you label both the receptor with the bone marrow toxin and of course the GFP as it's always present, when you increase the calcium, the red signal goes away and the green just gets enhanced, suggesting that there's those red signal never comes back throughout the experimental period, suggesting it's not a rapid recycling that's ongoing to increase those receptor levels. It's literally an increase from, or a trafficking from internal stores. Next slide. So finally, like calcium synthesis receptor is also involved in the non-calcitropic actions. So, up to now, there's been many different tissues that it's been, or other different types of tissues that it's expressed in, and it's demonstrated with different functions, such as in the brain and ischemia, as well as breast cancer and other colon cancer, to either increase or decrease metastasis, as well as in heart and so on. Next slide. One aspect that was also investigated was knockouts of the calcium synthesis receptor on bone development. So since it was been shown to be expressed in bone osteoblasts, and as well growth plate chondrocytes, the study was to see if a knockdown of this receptor had any effect on development, and clearly it did. We saw, it was observed that knockout reduced the bone growth and also affected the growth plate development. However, what wasn't or has not been fully investigated was whether it affect other cartilage or other musculoskeletal joints, and not just in the growth plate, but literally other cartilage sites. So could it be involved in degeneration or disease like osteoarthritis or so on? That hasn't been fully investigated. So next slide. Oh, next one. So to come to that point, we know that osteoarthritis is typified by an imbalance in cartilage formation, or homeostasis, form generation and degeneration, and that's what it's typified by. And in that process is so how that is involved is usually you have the implication of pro-inflammatory cytokines as well as upregulation of these catabolic enzymes, and during that change in milieu in the joint, you also see a downregulation of the matrix proteins that are essential for maintaining cartilage, which is the proteoglycan aggrecan and typically type 2 collagen. But what has been more recently discovered was that in the synovial fluid of patients with OA, calcium crystals were observed. And in fact, calcium crystals was proposed as a means of staging the OA disease. But the issue, well, the question it remains, I guess, is how these calcium crystals form, and are they, if they are to be used as, let's say, as a grading scheme for OA, are they inert, or can they also participate or in the degeneration of the cartilage itself? Next slide. What we found was that when we sampled some synovial fluid from OA donors in our laboratory, we noticed that the calcium, so the ionic calcium levels in the synovial fluid were higher than what would be predicted for normal. So normal in serum, for instance, in your blood, it's tightly regulated between around 1.1 to 1.3 millimolar. In the synovial fluid, that level is a little higher, but it's within that range, maybe up to 1.5. But what we found in at least 7 donors that we assayed, those levels were in the 2.2 millimolar. So the question was, can this calcium increase or be implicated in cartilage degeneration? Yeah, sure. Yeah, next slide. We also, since we had calcium, we found calcium in the synovial fluid. It was also important to make sure that the calcium sensing receptor was expressed if we were going to measure any effect on to make the association on how calcium could be inducing these changes. So what we found was that yes, a calcium sensing receptor in normal cartilage is very low. It's expressed, but the quantities are quite low. It's the Western blot on the right-hand side. When we looked at OA donors, we noticed that the levels were elevated, and this is immunohisto demonstrating the calcium sensing receptor in the corner there. And we also in these tissues, what is also typified is you see upregulation of pro-inflammatory cytokine IL-1. As well as COL10, which is implicated in calcification. Next slide. So as I mentioned earlier, there are many signaling pathways that are elicited by the calcium sensor receptor. The one I just want you to pay attention to or focus on is the MAP kinase pathway. Now, this is typically used as like a quick means of measuring for calcium sensing receptor activity, as opposed to the more complicated, detailed calcium imaging and so on. So MAPK is a good indicator of calcium receptor activation. Next slide. So we use this MAPK signaling, essentially just looking Western blots for ERK1/2, and we found in normal chondrocytes, these are normal human chondrocytes, you do see an increase in the MAPK signal with increase in calcium dose. Sure. But what we found was that in OA, that signal was, it was more potent, highly more potent. But regardless, even when you look at the dose response curves, even though the EC50 falls around the same dose, just the response was more potent, and it had to do largely to the probably expression of the receptor in the cells. Next slide. So we went on to do some gene expression studies with the OA chondrocytes. Next slide. And we found that if we incubated calcium, or if we added calcium sensor receptor agonists, because they do exist, along with antagonists are available for use. We found that calcium, particularly the agonist, was able to upregulate COL1 and decrease collagen 2 expression, whereas the antagonist reversed that effect. And this was somewhat comparable to the effects of the pro-inflammatory cytokine IL-1. The next slide. We also looked at other catabolic enzymes, and we found that similar to IL-1, calcium and the agonists, or activation through an agonist, which was calcium independent, induced the upregulation of agrokinases at MTS5. And next slide. And finally, in all cases, well, in either calcium or the agonist, although there was some variability with donors, we did find that alkaline phosphatase expression was elevated. Next slide. So in summary, we could say that COL1 was significantly upregulated by calcium sensing receptor at MTS5. And we found that the alkaline phosphatase, which indicated in calcification, was also upregulated. Next slide. We also went on to do some explants, so OA explants. And again, we use these osteochondral explants. We also noticed when we incubated them with calcium or the agonist, we saw decreased proteoglycan in the tissue, as well as decreased collagen content. But this was reversed when we added the antagonist in the presence of calcium. So we're able to inhibit the effect of calcium by introducing a calcium sensing receptor antagonist. And next slide. And one of the interesting aspects we started noticing was that if we incubated the cells with calcium for a period of time, 6 days, we removed the calcium over the course of 6 or 9 days, we started seeing a recovery of the expression of these matrix proteins called 2 and IRCAN. So suggesting that there is a reversibility if you inhibit the receptor or remove the agonist. Next slide. So that's pretty much what we're summarizing. So it's possible that regulating calcium levels in the synovial fluid or inhibiting the activity of the calcium sensing receptor may offer additional therapeutics for treatment of OA. So next slide. So other musculoskeletal disorders include the chronic back pain and the intervertebral disc. So intervertebral disc is one of these factors that are important not just for maintenance of the spine, but either through some abnormal means or through aging itself. When the disc does degenerate, and when upon doing so, this can cause issues with pain, compression of nerves, or just pain just through the disc itself. It's called discogenic pain, where you get a recruitment of nerve, you get hyperinnervation and pro-inflammatory cytokines and so on that just exacerbate a pain response. Next slide. So the structure of the disc itself, you have a vertebrae adjacent, two adjacent vertebrae, that, along with a thin layer of cartilage right before you, that's kind of sandwiched the intervertebral disc. And the disc that's sandwiched by these two vertebral segments is composed of an NP, which is a gelatinous center, which provides the hydration and compressibility, and an AF, which kind of supports all this and keeps it intact. So without the AF, for instance, the NP would just like squeeze and push out. So it's important to have everything intact. The next slide. The disc itself is kind of like glued or tethered to the bone through the cartilaginous end plates. And you can see here, you could appreciate in this H&E staining how the fibers or the cartilage kind of integrates with the part of the bottom part or the portion of the intervertebral disc, and as well as the bone. Next slide. Now, I mean, there's several causes of intervertebral disc degeneration, but the one that I'm going to have you focus on is the end plate. So the end plate, in addition to providing some structural glue or tether for the disc. It also regulates nutritional diffusion. Next slide. You could imagine that the end plate is kind of another barrier between the blood vessels that come from the bone and reach the level of the disc. So the disc itself is avascular, and its nutritional, the way it receives nutrition is literally through diffusion through the end plate. So if Misha, I don't know if you could just press.
I'm sorry.
Yeah, so yeah, sure. So as you can see, nutrients come through the end plate and provide some sort of food for particularly the NP as well as the NRF. And when this becomes calcified or degenerative, Misha, you can just press. This prevents or limits nutrient diffusion, and which of course could lead or exacerbate the degeneration of the disc. Next slide. So we were interested in since we've already started, we were investigating the cartilage, and since of course cartilage's end plate is important for nutrient diffusion, we started looking at aspects of this end plate in the human disc. And as you can see here, alizarin red is a means of staining for calcification, and the cartilage end plates that we extracted from donors, we found that with increasing degeneration, which is the grading scale, we found increased calcification, shown there, grade 4. This was also associated, as expected, decreased protoglycan content, as you see there in the bottom left hand, and collagen content as well seem to be markedly decreased in these degenerative end plates. Next slide. Now, calcification, if this is, since we've observed it with degeneration in the end plate, it needs to take place. You need phosphate, of course, to induce to form a calcium crystal, as well as alkaline phosphatase. So you need like high levels of calcium and phosphate for this to occur. So next slide. So what we did was we measured the calcium content in the end plates from these degenerative tissues, and we found that there was a significant increase of calcium in the end plates of highly degenerative discs. So the question was is that implicated in the degeneration process, or can it participate in? Next slide. And so we isolated chondrocytes from the end plates. We incubated them with different concentrations of calcium. We also incubated interleukin-1 beta as a control, as we knew, as it's known that interleukin-1 can regulate the expression of amino proteins in chondrocytes. And what we found was that calcium induced a very nice dose response on the expression of COL1, COL2, and aggrecan in these cells. It's Western blotting, and what was nice as well was that the dose response effect was occurring, as you see here in the right hand side, the EC50 for aggrecan and COL1, 2. So it's between 1.5 and 3 millimolar. So it's within like it didn't take a lot of calcium, to start seeing changes. Next slide. So we also looked for the calcium sensing receptor, since we noticed that that curve that you saw was around 3. So the EC50 of the calcium sensing receptor, as we observed and others published, is around 2.5 millimolar, right? 2.5. So the effect was occurring within that range with the end plate cells, so which led us to believe that it was a calcium sensing receptor response. And as you could see, when we stained when we did immunohisto for calcium sensing receptor, it was upregulated with degeneration and by Western blot by immunohisto. Next slide. We then incubated the cells with antagonists. So if we could can we inhibit the calcium sensing receptor chemically? in the presence of calcium, would that affect it? And yes, it did. So we were able to reverse that downregulation of those matrix proteins in the presence of increasing antagonist. Next slide. And finally, yeah, we also demonstrate that it not just inhibiting, but if you actually activate it allosterically, so you don't add calcium, you just add an agonist that makes it more sensitive to low calcium, which was a 1 millimolar. And by just doing so, we were able to see decreases in collagen and aggrecan. Next slide. So we went on and we did organ cultures. So these organ cultures is where you actually remove the bone, and we found that when you increase, so when you in the bath that you culture these organs, these are bovine organ intervalist organ cultures. When you increase the calcium in the bath, and this is a 2.5 millimolar, and look for it and look at the histology of the end plates, we found that there was calcification by von Casa, and there was increase in alkaline phosphatase, as shown there. When you maintain the calcium levels at 2.5, but you put inhibitors of the calcium sensing receptor in the bath, that decreased that effect. Next slide. We also looked at proteoglycan levels, so you could partially restore the proteoglycan levels with the antagonist in 2.5 millimolar calcium. As well, we had strong recovery of collagen in these organ cultures in the presence of the antagonist. Next slide. And we also did knockdown studies. So if we isolated the cells, incubated them with siRNA, there's a control and calcium, anti-calcium siRNA, and we were able to show that in the presence of calcium, you could reduce or recover, or completely obliterate the effect of calcium on matrix protein expression in these cells. Next slide. Surprisingly, there were no changes in the enzymes. So aggrecanases, MTS4 and 5, and MMP, which is a collagenase. Next slide. But calcium itself actually was able to activate enzymes implicated in proteoglycan degradation. So when we took purified aggrecan and took conditioned media from end plate cells that were conditioned in either 1, 2.5, or 5 millimolar calcium, added that conditioned media to purified aggrecan, we were able to see almost complete loss of the aggrecan. This is by Western blot on the left-hand side. And we observed that using ADAMTS5, the enzyme itself, just incubating the enzyme in the presence of purified aggrecan with increasing calcium concentrations, you can increase the enzyme activity. So, and these enzymes are sensitive as well to calcium levels. Next slide. So the last thing we did was we looked at nutrient diffusion. So if the end plate is important for nutrient diffusion, as mentioned, and calcium was perhaps inducing calcification as well as degeneration of the end plate, as we observed. Then under high calcium concentrations over a period of time, that should block nutrient diffusion to some extent. And using glucose analog, we were able to observe, we should just press it again, you'll see. When we increase the calcium concentration, we saw decreases in the glucose diffusion. However, when we added the antagonist in the presence of the elevated calcium, maybe you could just press, we saw a restoration of the glucose diffusion. So next slide. So as it summarize, so calcium is elevated in degenerative discs. From the end plates of degenerative discs, it does regulate matrix protein synthesis. This happens through the calcium sensing receptor, and this could impair an activation of which can impair glucose availability or nutrient diffusion. Next slide. So the next, well, kind of the final question was whether calcium could be implicated directly in degeneration of intervertebral disc. Up to now, it was just the end plate, but can it affect also disc tissue? Next slide. So we know, based on X-rays from patients' spines, that degenerative disc can get calcified, and sometimes quite severely. You see actual large calcium deposits. That's what's shown here in this X-ray radiograph. Next slide. And then our lab's also shown by histology that you get through von Casa staining that various aspects of the disc, the NP, the center, the AF, the outer, as well as the end plate itself, can undergo calcification or demonstrate mineral deposits. Next slide. Now so the disc matrix itself is regulated by the cells in question. If we focus on the center, the nucleus pulposus, which is the major part of the disc, it's like always in a homeostasis. So there's always a level of synthesis and degradation, and during disc pathologies, it's usually the balance is towards degradation. So you see loss of proteoglycan and collagen synthesis, as well as an increase in their degradation. Next slide. So what we did was now continuing from the end plate, we measured calcium levels in the disc itself. And what we found was that there was a nice correlation between severely degenerative discs and increase of calcium content in those discs. Next slide. We also looked for the calcium sensor receptor expression. This is histology showing in grade 2 and grade 4 comparison that you see increased presence of the calcium sensor receptor in cells of those degenerative discs. Next slide. We took the NP cells, the human NP cells, incubated with different concentrations of calcium, as well as the calcium agonist I mentioned, the allosteric agonist and the calcit, and we compared everything with interleukin-1, known factor that's implicated in degeneration, and we saw that with increase in calcium, you see comparable effects to our pro-inflammatory cytokine control IL-1 on collagen, as well as aggrecan expression in these cells. Next slide. To answer the question more, I guess, about whether calcium is implicated in the degeneration of the disc itself, we developed a rapid means of isolating vertebral segments from bovine tails that contain bone, so we don't have to remove all the bone itself, as we did in the previous studies. And this was a fully intact disc. So if calcium was going to come through, it would have to penetrate through bone, right through, and then through the end plate into this tissue. Next slide. Next slide. So in the past, like I mentioned, we had to remove that bone because it was interfering with cell viability when in culture for extended periods of time. Next slide. But with our media system, so with our system, we were able to maintain that bone and allow the discs to stay viable in culture. Next slide. This is a quick way of how we do it. It's kind of messy. You're literally like in a butcher shop. So you have to like remove all the tissue and cut the bone into segments, clean it up, and then through the media system, you result in these nice discs as shown there on the bottom right. And we culture them in these little cups with filters. Next slide. And this is just a system that it's like a 3-step system. And you see the nice intact disc when you're done at the end of culturing. Next slide. It is all demonstrating the viability. So you do maintain high viability in all compartments of the disc after isolation. Next slide. As well as glucose diffusion, which is the important aspect. So the nutrient diffusion is maintained in our system. Next slide. And proteoglycan content is just further demonstration that the disc is healthy and intact. Next slide. And this is the, so one of the things we want to determine is, can calcium itself induce cell death? Because it's possible that you know, just those levels alone may be inducing cell death, but surprisingly it was not. So even up to 5, even if the discs were in baths of 5 millimolar calcium, which would be high for cells, there was no observable decrease in cell viability. Next slide. However, we did see decreases in the proteoglycan content of in NP and AF tissue, and this was also evident even if the disc, even if the environment and media was maintained in low calcium, say 1 millimolar. If you put the antagonist in there, which is the allosteric agonist, the allosteric agonist, you were still able to induce those changes as though the calcium levels were elevated. Next slide. And this is to show the staining. So this is another means of showing proteoglycan content in this tissue. It's a sapho staining, and you can see it goes from red to very pale to faint as you increase the calcium dose. And you saw similar change. We see similar changes in the staining when the discs are just cultured with the allosteric agonist and the calcium. Next slide. And this is demonstrating the collagen content. So we were able to also see decreases in collagen content in the tissue. Next slide. And finally, in collaboration with Biomomentum, using their MACH1 system, we did a 1 month, this was like a 4-week study, disc culture with either calcium or 2.5 millimolar or cinecalcit in 1.0 millimolar, so in control media. And what we observed was that the structural stiffness of the tissue, NP in particular, there was increased stiffness in the NP tissue in these organ cultures. There was differences, there were slight shifts you could see in the AF with 2.5 millimolar, but it didn't look like that was significant, but in the NP, we definitely saw differences. Next slide. So we can summarize that calcium suppresses aggregate and collagen expression in disc cells, and this is largely through the calcium-sensitive receptor. It doesn't affect cell viability, and it does increase disc stiffness. Next slide. So so regulating the receptor may not only improve disc nutrition, but also may mitigate a degeneration as well. And so the point here is, especially when we're treating disc with all these biologics that are in development or being investigated, if the environment of the disc is already poor and the calcium levels, as we see, are already elevated, it might be something to consider when you're trying to gauge your treatment, because if the calcium levels are still there, you don't find a means of regulating this, you know, your biologics may not be very effective. Next slide. I'd like to thank investigators, so it's Dr. Mola and Antonio, a lot of my work in vitro studies that students that helped me out were Yazid and Rakan. And of course, the biomechanics studies were done by Biomomentum, and the support was through CITAR and AO and NAS foundations. So thank you for your attention. Free to ask if we have time to take any questions.
Thanks, Michael. Thanks for speeding up. No, it's not easy. So that actually was my question, but let's see if anyone in the audience has any questions. Thank you, Elaine. Do you have any questions? So yeah, I mean, Michael, when the reason why calcium levels are quite tight, I thought, in the body was because once they go above a certain level, you go beyond the sort of solubility limit or whatever of the mineral, and mineral is precipitated, right?
You can't, so in patients who are FHH, so those who have mutations in the calcium sensor receptor, free calcium, so not bound, because bound is almost double, right? You go 2 to 3 millimolar, I think, or 2.5 millimolar if you're talking about total calcium. So proteins do absorb some of that calcium. The free is, yeah, like you said, it's tight, 1.1, 1.3. The crystallization, if we're talking about, yes, you do get ectopic crystallization if it does go to a certain point, but in these patients, you can see it up to 2 or more free calcium when uncontrolled. So
Yeah,
it's not just, so they can go,
yeah. Yeah, it's high. So in normal people, it doesn't go high. So why is it high? Where is it coming from?
Bone and loss of, I mean, the hormone PTH is what regulates it. So if you don't excrete it properly, and if your bone is constantly being resorbed, then that's what's leading to the increased levels. There is gut as a factor, so vitamin D is also increased when calcium levels are low, and vitamin D, of course, helps with calcium absorption through your nutrient intake, but it's mostly through PTH.
So the calcium comes from the bone.
Yeah.
But the bone's not calcium, is it? It's calcium and phosphate. So
yes, hydroxyapatite.
Wouldn't you raise both those?
Yes, you actually do. Yeah. So what happens with the kidney regulates the phosphate. So to try to maintain calcium levels, phosphate's excreted. If it's resorbed when you try to decrease serum calcium levels to balance the calcium, yeah, to keep it inactive.
To me, are these patients their calcium's elevated systemically or just locally to the area of degeneration?
So in the mutation ones with the variants, it's systemic. Yeah.
And you something that annoys me that biologists do is they talk about calcium crystals as if such a thing exists, right? So, you know, I believe the crystals you're talking about are normally pyrophosphate, hydrogen phosphate rather, or urate.
Yes. It could be a multi. Yes.
Is there any difference, or does it matter, or?
You know, yes, I believe for the calcium crystals, some are can induce different, depend on the type of crystal in order to have a response. So those were other investigators looked at the effect of calcium crystals, but the actual effect of free calcium, that's not something people have really looked into. So you can have calcium crystals, but you can also have free calcium that's there, that's present, that's unbound. That could be doing something.
We have a question from Elaine. Would taking calcium supplements help, or are even suggested after a certain age? Or is that choice to take supplements daily in fact dangerous if it reduces vitamin D? Would it be important to reduce calcium intake supplements and take more vitamin D? There's quite a few questions there. So,
I think at one point, like, vitamin D was considered a super vitamin, and I think the conferences I remember going to, people were promoting it, like as though everybody should be heavily dosing themselves on a regular basis because it could cure some sort of multitude of things like cancer and so on. I think now it's becoming clear that too much calcium, too much vitamin D is problematic over the long term. I mean, at some you do have to maintain your calcium levels. You do have to get it from food intake. And if there is an imbalance, what happens is it kind of sets off like issues like kidney issues. Secondary hyperparathyroidism is another problem that occurs in chronic kidney disease. Imbalances in calcium could also cause problems to homeostasis because you activate PTH and so on in a sustained manner. So it has to be tightly regulated. Too much is no good. It's like a sweet spot. You just have to maintain the levels that are recommended.
So moderation, I guess, is the
Perfect. The calcium.
Couple more questions for you then. So if the use of calcium, I believe you were saying calcium receptor antagonist is effective in reducing at least some of the markers of degeneration.
Mm-hmm.
But is taking that systemically viable?
For certain, like for patients, for FHH, for instance, that they have a calcium disorders, they do take a drug is cinacalcet. So the drug I used in the study was the one that's actually given in the clinic. For the antagonist, I think it hasn't really been sufficiently investigated for systemic use. I would imagine a local delivery would be appropriate because systemic, of course, you're gonna raise the PTH levels because you're inhibiting the receptor. Yeah.
So your vision is the pre-osteoarthritic patients would show up for like joint injections. Is that what you're saying?
Yes, exactly.
But how do you know which joint? I mean, it's the whole spine, right? It's a lot of joints.
MRI for the joints would help you determine.
Has there been any study to show that localized antagonist delivery prevents the onset of osteoarthritis?
No, actually, not that I'm aware of.
Isn't that what kind of your work's leading to?
Yes.
Yeah.
Yes.
Okay.
It's something that we'd like to do, animal studies.
Yeah, sounds fiddly. Because I think there are a lot of injections, aren't there, in knees and hips that may or may not do anything, right? These hyaluronic acid injections and okay. I don't know very much about the calcium receptor other than it's a promiscuous receptor, and any divalent cation will activate it. Do you have any comments on magnesium?
Oh yeah, it does activate it to a lesser extent. Maybe I think you have to reach up to 20 millimolar to start showing some EC50 comparable to 2.5, about 10-fold less. But it can activate, yes, as well as some antibiotics. Even acidity could also trigger act as an allosteric regulator. You could trigger activation. There are several factors that could activate or push it towards a more sensitive or active state.
Okay, that's great. Do we have any other questions for Michael? If not, then thank you very much again for showing us your very interesting work. And congratulations for punching through the IT problems, and thanks everyone for their patience. And good luck with your research. Thank you.
All right, thank you.